Okay, so today we're going to go over AP Biology Unit 2, Cell Structures and Functions, the quick review. Alright, so the way we're going to structure this video is basically College Board has set up certain units for each, certain subunits for each unit, and we're just going to go through each and basically explain them. Alright, so first one, I've decided couple 2.1 2.2, so cell structures, subcellular components, and function. So let's look at the difference between prokaryotes and eukaryotes. So just some general differences is that prokaryotes don't have any internal membranes. They don't have any nuclear membranes, endoplasmic reticulum, mitochondria, vacuoles. Alright, so they have circular, naked DNA. This means they're not bundled or wrapped in any histones or chromosomes. The ribosomes are very small. Metabolism is anaerobic or aerobic. They have no cytoskeleton, and they're mainly unicellular. There's very small cells, 1 to 10 micrometers in length. Eukaryotes, on the other hand, contain distinct organelles. They have DNA wrapped with histone proteins in chromosomes, larger ribosomes. Metabolism is aerobic or uses oxygen. Cytoskeleton, multicellular with differentiations, which basically means that cells will adapt as need be, and they're larger cells, 10 to 100 micrometers in length. So this is a quick diagram of the eukaryotic cell. As you can see, there's rough endoplasmic reticulum, there's lysosomes, vacuole, nucleus, chromate, and so on. So let's get into it. So let's look at a few cell organelles in particular. So nucleus. It contains chromatin that's compressed to chromosomes surrounded by a nuclear envelope, and it contains pores only RNA can pass through. That's important. RNA, in translation, only RNA can pass through. DNA has to stay within the nucleus. It is the site of cell RNA production. Nucleolus. This is a part inside the nucleus, and it is the site of ribosome assembly, and there could be multiple depending on the cell type. Ribosomes. Translates RNA sequences into polypeptide chains, and it contains larger and smaller subunits, EPA sites, and then attached to rough ER or free floating. When we learn about transcription, we'll look into EPA sites a little bit more deeply, but this is a general statement of what ribosomes are. The endoplasmic reticulum. This is interconnected membranes of channels and flattened sacs. Interior lumen is very different. There are two types of endoplasmic reticulum. This is shortened to ER. Rough ER, which has ribosomes attached to produce proteins. So the entire point is to use those ribosomes and produce proteins for the cell to use. The smooth ER assists in hormone synthesis. It stores calcium plus ions in muscle cells for normal muscle contraction. It detoxifies drugs and poisons from the body and is a site for glycogen degradation. The Golgi apparatus. This processes and packs proteins for transport. It's like the mailman of the cell. It secretes to other parts of the cell or to the surface for export. It adds carbohydrates to proteins and creates polysaccharides for plant walls. And if you want to see here, the nucleus of course is centered right here. The nucleolus is within the nucleus. The ribosomes, as you can see, are the black dots, and the rough ER is loaded with ribosomes that are attached to it. Smooth ER is shown right here. Then we have the Golgi apparatus, which is right here. And this will transport materials throughout the cell. Okay, so the mitochondria, as everyone knows, is the powerhouse of the cell. It breaks down energy-rich molecules into ATP. It has an outer membrane with pores and inner membrane separating biochemical processes. Lysosomes are sacs of hydrolytic or digestive enzymes. It helps digest molecules. Phagocytosis, it's a way lysosomes can it forms a pocket around a macromolecule. This is called a phagosome, the pocket with the macromolecule, and it combines with a lysosome to form a secondary lysosome to digest the material. So to get this right, there's a lysosome and there's a phagosome. When they bind, they create a secondary lysosome. Since the lysosome contains digestive material, this digestive material will break down whatever molecule that is, and then the cell can proceed to excrete its waste or use it in some processes. Autophagy is a programmed cell death where lysosomes pop and digest everything. If it fails, many cancers form this way. Autophagy, the lysosome, which contains digestive materials, actually pops, and this digestive enzymes are then spread throughout the cell, destroying the cell. However, if autophagy cannot occur, then cancers form. Cancers replicate without any restrictions. So if a cell cannot basically program its own death, then it will continue to replicate, and that mutated gene will continue to spread. Perioxysomes contains catalase, which breaks hydrogen peroxide, which is toxic to the body, and detoxifies alcohol. Glyoxysomes are lipids converts lipids to carbohydrates for growth. And the cytoskeleton is a complex of protein filaments throughout the cytoplasm. The main purpose of this is to maintain cell shape, contain positions of organelles, flow of cytoplasm, cytoplasmic streaming, and anchors the cell in place. It's consistent of microtubules, which are hollow tubes of protein tubulin, spindle fibers, which separate chromosomes, microfilaments assembled from actin filaments to support shape and enables cleavage for cell division, and skeletal muscles to contract as they slide along myosin filaments. Okay. We still got more. Vacuoles. So vacuoles are both in plant and animal cells. However, they're much bigger in plant cells, and they have more purposes. So large vesicles, or many small, like in eukaryotes, functions of, so they store toxins and waste for later excretion. They help in structure. When water is put into the vacuole, it will expand, causing the vacuole to inflate. And for plant cells, this will allow increased turgor pressure and rigidity along the cell walls. So it'll actually help the plant stand up straighter because it puts more pressure on the walls, causing them to stand straight up. Reproduction, there's pigments that attract animals within these vacuoles, and those animals will help spread pollen and seeds. Catabolism, it hydrolyzes proteins in monomers for plant seeds to use. Alright, so next is chloroplast, another commonly known organelle. It's just a pigment inside the plant, and it's the site of photosynthesis. It has a double membrane, which is due to the theory of endosymbiosis, and we'll explain that later in the origins of life. But yeah, it has a double membrane and thylakoid stacked in grana. So cell wall. Cell wall is also another thing that's only found in plants, and it's made of cellulose in plants and chitin in fungi. It protects the cell and keeps shape. Plasma membranes, which is a big focus of this unit, is a phospholipid bilayer surrounding the cell and determining what comes in and out. It's formed of phospholipids, and basically this will protect the cell from and allow the cell to transport materials in and out to its environment. And then finally, the cilia/flagella is made of microtubules that allow the prokaryotes to move. They're made of microtubules, and they allow it to move in any substance or situation. Okay, so cell size. This is very simple. Cells. Why do cells divide? Well, cells can't get too big due to surface area to volume ratio. If volume grows faster than surface area, then it won't work, because it needs surface area to actually transport materials from the outside to the inside. If there's too much volume and too little surface area, the cell won't be able to transport all the nutrients it needs in order to actually survive. So by dividing, it'll increase the surface area, allowing more transportation and easier, and basically it'll allow the cell to move materials wherever it needs to be. As you see here, I know cells aren't cubes, but this is a good example of why we divide. So in this case, this cell has a 1 by 1 by 1 ratio. There's like 1 by 1 by 1 dimensions, and when multiplied, you see, so 1 times 1, this surface area is 1 centimeter cubed, and if you times that by 6, it will be 6 centimeter cubes, compared to the area, which is 1 times 1 times 1, so it would be 1 centimeter cubed. Now, that's a 6 to 1 ratio. The surface area is 6 centimeters squared versus the volume, which is 1 centimeter cubed. Same thing here, and we see a 3 to 1 ratio, which is a much lower ratio than 6 to 1. And then finally a 2 to 1 ratio. At this point, the cell cannot possibly function properly and needs to divide. Alright, plasma membranes. So, the plasma membrane separates the cell from its environment. It's constituted of lipids, proteins, and carbohydrates. Carbohydrates are used for signaling, but we will not have to cover that for the AP Bio test. So, phospholipid polarity interacts to form bilayers. Phospholipids have two sections, polar and hydrophilic ends, phosphorus, and nonpolar and hydrophobic ends, fatty acids. The proteins embedded in the bilayer they move materials through the membrane, intracellular recognition and adhesion. They receive chemical signals from the environment, and the holes in the membrane are automatically sealed themselves. And as you can see here, this section right here is the polar hydrophilic ends, and then this green section is the fatty acids. The more saturated a fatty acid is, or if a fatty acid is saturated, there will be actually less fluidity in the membrane, and we'll see that soon. So membrane permeability, what affects how fluid a membrane is? Membrane permeability is actually the ability for materials to transport in and out through the membrane. So things that affect fluidity are the lipid composition. So for example, cholesterol, more cholesterol, longer fatty acid chain, and increased saturation will lead to tightly packed phospholipids and less fluidity. Short fatty acid chains plus unsaturated fats, they increase fluidity. Temperature, decreased temperature means less fluidity within the membrane, and outer and inner layers may have different compositions. Animals can change their composition due to their environment. So this is interesting because animals, depending on their environment, if it gets cold or in the winter, they can actually tighten their, membranes by increasing cholesterol counts, and this will lead to less heat loss. Also, neurons, a good example of this is anesthetics. When anesthetics are applied, neurons actually harden or they become less fluid, and this kind of blocks the transmission of a signal. So when the neuron is sending out a communication signal, it will actually not be able to be received, causing a numbing sensation. Okay, membrane transport. So proteins that assist in transport. There's hydrophobic and hydrophilic R groups that determine the protein's position and insertion. So the types of membrane proteins: peripheral membrane proteins. There's no hydrophobic regions, so they're attached on the outside. Remember, the fatty acids are hydrophobic, meaning if there's no hydrophobic region, it cannot be within the membrane. Integral membrane proteins. They have both regions, hydrophilic and hydrophobic, so they're partly or completely inserted depending on the location. So two types: anchored membrane proteins, proteins with lipid ends that anchor them into the cell membrane, and transmembrane proteins, which span the entire length of the membrane and have proteins sticking out on both sides. Alright, transport. So membranes have selective permeability along with only some molecules to pass through. So some by concentration gradient and others by special membrane proteins. So the first type of transport we'll look at is passive transport. Passive transport requires no energy, hence passive, due to concentration gradient. Things will move just due to how the concentration gradient is aligned. Diffusion is an example of passive transport and is the process of random movement to equilibrium. So the higher concentration gradient and the lower concentration gradient, the molecules will constantly be moving between them until both gradients are equal. So speed of diffusion is dependent on the diameter of the molecule that's being diffused. Temperature, if there's higher temperature, that means there's more kinetic energy, meaning that the molecules are moving faster and they'll quickly get to the spots they need to be. And then concentration gradient. The greater the concentration gradient, the more movement there'll be. So small nonpolar uncharged molecules move through the membrane through simple diffusion. They pass, so this just means they go through easily. Large, charged, and polar must be aided by protein channels. These molecules cannot pass through the membrane and need protein channels, as we talked about earlier, to get them through. Alright, active transport we will talk about later. So, facilitated diffusion. We talked about simple diffusion here. So what is facilitated diffusion? Protein channels aid in diffusion. So there's two types of proteins that help with facilitated diffusion. Channel proteins, which form channel across, and carrier proteins. The channel protein is shown right here, and a carrier protein is shown right here. So a channel protein. There's two examples: ion channels. These are gated channels. This gap right here is closed before a ligand binds, and when a ligand or a signaling molecule actually binds to the receptor, to the channel opens up, allowing the molecules to pass through. Some ion channels actually open to charge differences. So if the outside charge is different from the inside of the cell charge, then these channels will open up. Okay, aquaporins. So basically, water can actually diffuse through the membrane on its own, but very slowly. It can also actually link onto ions while they pass through ion channels, and up to 14 at a time. However, aquaporins, there's special channels just for water to get through, and if a cell opens up, water can quickly pass from one area to another. So anyway, next is carrier proteins. Carrier proteins is shown here. This will actually work it changes shape upon binding of a polar molecule. It releases molecules in the cell or out, and the cap speed, so when it reaches the most it could transport, is called saturated. Alright, tonicity and osmoregulation. So, osmosis. Osmosis means it is water moving down a concentration gradient through diffusion and aquaporins. Osmotic pressure is pressure needed to be applied to a solution for the flow of water. Turgor pressure is internal pressure on cell wall, and it maintains structure and stops the flow of water. There's an equation for this soon that we'll learn when I create a video on the equations. It actually allows you to calculate osmotic pressure, and you can predict the flow of movement of water. So some terms used for comparing solution is hypertonic, which is solutions with greater solute concentration. This leads to plasmolysis in plants. There's hypotonic, which is solutions with lower solute concentrations. Cells will actually swell and become turgid. Lysis will occur in animals. And isotonic, solutions with equal solute concentration. So if a cell is hypertonic to its environment or its surroundings, then that means that the concentration inside the cell is higher. So water from the outside will actually flow into the cell, causing the cell to grow or swell up. Hypotonic means that the cell has less concentration than its environment. This means that water will actually go out of the cell in order to balance the concentrations. Because remember, concentrations always want to reach isotonic. Isotonics means they're equal, meaning that the net flow of water will be zero. This doesn't mean no water is flowing. This just means that the water moving in both directions cancels each other out, and the net concentration or net movement will be zero. Osmotic pressure. Osmotic pressure is the tendency of water to move across a permeable membrane. And lastly, water potential. Water potential is potential. The water potential depends on solute concentration and pressure. The water potential of pure water is zero. This is because pure water has no solutes in it, causing it to have zero water potential or zero gradient, zero movement. Anyway, mechanisms of transport. Here we go. Passive transport we already talked about, along with simple and facilitated. And then osmosis we talked about in the last slide. If you want to read over that real quick, pause the video. So active transport. This is the movement of molecules against a gradient. So pumps/carriers carry proteins across the membrane by active transport. The sodium-potassium pump pumps sodium and potassium ions across the membrane, returning the nerves in a neuron membrane, electron transport chain. It pumps proton ions across inner membrane. Exocytosis. This uses vesicles to release particles. In neurotransmitters, it releases neurotransmitters into the synapse. Pinocytosis is cell drinking. It's an uptake of large dissolved particles by forming a vesicle with the cell membrane. Phagocytosis is engulfing large particles or small cells by membrane wrapping around particles and enclosing it. It uses pseudopods. Pseudopods is, in essence, when a cell is taking up a molecule, it will form this kind of bend or curve. This just to take up the molecule, which we'll work on later. Both of these areas, are called pseudopods, and eventually it will engulf the entire molecule it's trying to surround, like that. Okay, that was very crappy drawing, but I hope you understand what I was trying to say. Okay, so receptor mediated endocytosis. There are receptors that line the cell membrane, and the cell takes up large amounts of specific substances. These substances will bind to the receptor, and then once they bind, the cell membrane would actually fold in on itself and take up the molecules that it needs. So once a ligand binds to the receptor's membrane, endocytosis begins, and a coated vesicle forms. This is common with cholesterol. And bulk flow is the general flow of a fluid, such as blood in humans. They always flow in one direction and for a specific purpose. Alright, lastly, 2.10 and 2.11. Origins of cell compartmentalization and what it is. So basically, cell compartmentalization is just the separation of jobs throughout different organelles in a cell, and this causes the cell to work more efficiently when separated. The theory of endosymbionts, I can't say that word. This theory, as I explained earlier, it basically, ancient times, a cell, they thought that chloroplasts and mitochondria were actually separate. They lived on their own, and they were free-living organisms until they were taken up by a larger prokaryote, and it was beneficial. This symbiotic reaction or relationship was beneficial, so they kept it going. Some evidence that this is true is chloroplasts and mitochondria have their own DNA, suggesting that they were once their own organism. And DNA is more prokaryotic and not wrapped within both chloroplasts and mitochondria. And both of them have a double membrane. One is their own when they were living free, and another is of the cell when it was taken by endocytosis. Remember, the prokaryotic cell actually took in the chloroplast and mitochondria, creating another vesicle around it. Alright, Unit 2 is done finally. My throat is... Alright, anyway, thank you for watching, and I hope that helped.